Hi, this movie guides you about the activity of the Squalomix consortium and its recent progress. My name is Shigehiro Kuraku, professor at the National Institute of Genetics in Mishima, Japan, leading this consortium. This time, I'm recording this briefing to convey to you what is ongoing, whichever time zone you are in, and whatever piece of information you are interested in, so that you can watch this movie anytime you want. Shark research in my lab in Kobe was initiated around the year 2013, about 10 years ago. Since then, we have built up a network involving biologists, technical staffs, researchers, and animal caretakers in public aquariums. Basically, we are funded to conduct academic research projects to address biological questions, especially in developmental biology and physiology. We release data as a part of those question-driven projects run by individual students and postdocs, mostly working in my lab. This is why we may be sometimes slow in releasing data, I must say. We collaborate with researchers in other labs, sometimes in other countries, and also communicate with other parties affiliated with Earth Biogenome projects. To minimize overlapping effort, we are open to your ideas for collaboration on the topics we have not explored in the cartilaginous fish genomes. Here is a list of the parties, like aquariums and researchers collaborating tightly with us, and I would like to send my best gratitude to them. Initially, we were driven by questions in evolution biology, pursuing the vertebrate ancestor, but our motivation has been diversified. It now covers molecular level understanding of the difference from the rest of the vertebrates and the diversity among cartilaginous fishes. We are also interested in molecular basis of the remarkable phenotypic traits like sensory capacity and life history traits, including long life. In the long run, we expect that our activity also contributes to the maintenance or recovery of biodiversity and marine environment. Our activity is not limited to producing and analyzing sequences and includes establishing analytical tools and materials. What you see here is a batch of culture cells that were first used for karyotyping, and more recently, they are used for testing exogenous gene introduction, assuming future applications to operative experiments. Also, genome scaffolding can be better performed by using chromatin contact data obtained from a more uniform batch of culture cells than real animal tissues. Our activity has been primarily enabled by collaboration with experts inside my own lab, with the complementary skills included here. Even after our base is moved to National Institute of Genetics in Mishima, some of this expertise is further consolidated by new lab members. I also welcome contacts from overseas researchers seeking opportunities to work with us. Now I get down to briefly introducing our recent achievements. The first item is about vision. We focused on a light sensor in the retina, rhodopsin, also functioning in our eyes. We previously reported the spectral tuning of the whale shark rhodopsin to the wavelengths of light that penetrates deep water. Later, we newly designed an in vitro experiment using synthetic whale shark rhodopsin proteins. This identified two amino acid substitutions that enable the spectral tuning, which has never been documented for rhodopsin of any wildlife. One of these substitutions at the site 94 actually is also known to cause night blindness in humans. Whale sharks dive down to the deep ocean of 2,000 meters, although we do not know well what they are doing in the deep. Very interestingly, a whale shark has its own unique mechanism to sense the dim light in the deep, and we discovered this with in vitro experiments without sacrificing animals. The next one is about reproduction. We focused on the difference between oviparity, that is egg laying, and viviparity, namely live birth. An important piece of prior knowledge is that placental mammals, including us humans, lack egg yolk protein, vitellogeny, from the genome. We then examined whether live-bearing sharks and rays also lost these egg yolk proteins. We analyzed the accumulating sequence data for 12 shark and ray species and showed the persistent repertoires of vitellogeny genes in the genomes of sharks and rays. From this study, we learned that viviparity of sharks and mammals hold on a different background. In fact, many viviparous sharks and rays feed babies with yolk partially, and our finding is consistent with this. Another lesson generally observed on a genomic scale so far is that the sharks and rays have often retained very well gene repertoires that presumably existed in the common ancestor of vertebrates. The latest one is about shark chromosome evolution. Please note that this study was based on short read sequencing, not long reads, simply because we long struggled to publish this. We reported chromosome scale assemblies of the zebra shark and whale shark. Actually, the blood samples used came from the individuals still living in the aquarium tanks in Okinawa, thanks to enthusiastic cooperation from the Chiraumi Aquarium and their animal husbandry skills. In fact, VGP, Battery Genomes Project, also released the Hi-Fi long read based zebra shark genome assembly for your information. Using our products, we characterized the chromosomal sequences of a wide spectrum of lengths. This brought us to some interesting patterns. First, shark has a quite gradual distribution of chromosomal sequence lengths, and it is hard to divide into macro and micro chromosomes only by length. Among those chromosomes of variable length, shorter chromosomes tend to have higher GC content, shorter gene length, higher gene density, higher synonymous substitution rate, and higher frequency of simple repeats. Some of these characteristics were previously observed for microchromosomes of birds, like chicken. But we caution the use of the word microchromosome because of no uniform definition. It would be safe to use the word microchromosome only within a karyotype in comparison with much larger chromosomes of that species. This is a quite heavy slide, so if you feel unfamiliar, please feel free to skip this slide. Some studies try to find a conserved synteny in small microchromosomes as evidence of their common origin, but chromosomes intermittently undergo fissions and fusions. And probably there were times with the microchromosomes after fissions and times without microchromosomes after massive fusions. Both should be correct. These processes are reversible in a bistable manner, and there is no single answer like with microchromosomes or without. Still, we unwillingly grouped shark chromosomes by length, and as I said, we found certain tendency which roughly recapitulates what was previously shown for microchromosomes of birds and reptiles. Small chromosomes resemble the ends of larger chromosomes, and in every chromosome, the ends of certain lengths have such different characters from the rest. In here, the short chromosomes are more widely occupied by its ends, and consequently, short chromosomes as a whole look more high GC content and exhibit other characters I mentioned. We also discussed more aspects of chromosome organization, and we are happy to exchange more ideas with those interested in these topics. In the same paper, we also reported X chromosome. This is the first time in publication a shark's sex chromosome is characterized in support of its DNA sequence. Our identification was based on male-female comparison of whole genome sequencing depth with the single male-female pair in which chromosome 41 stood out. The result was confirmed by quantitative PCR-based experiments with 3 male and female individuals. In this synteny view, genes on zebra shark X chromosome showed orthology to human and chicken genes on their autosomes, not sex chromosomes. I did not show here, but we could not find any commonalities with the teleost species either. This suggests an independent origin of sex chromosomes between zebra shark and other species. We are extending this comparison to include more chondrichthyan species and also looking for Y chromosome. The last item I introduced here is about Hox genes. As I said earlier, shark genes tend to be well conserved, but among Hox genes, this trend applies to only Hox A, B, and D cluster, but not to Hox C. The Hox C cluster was previously suggested to be completely lost from the genome of sharks and rays. Despite this report, we showed the retention of Hox C genes and later the chromosomal linkage between them. Our comparison, however, revealed the possible difference in the repertories retained by different shark and ray species. In fact, the retained Hox C genes do not fit within 100 kilobases, the usual length of a Hox cluster in many vertebrates. And the cluster permitted massive invasion of repetitive elements, which is again rarely seen in vertebrate Hox clusters. This time, in our latest publication, we localized this unusually evolving Hox C cluster in the PAR, which is pseudo-autosomal region of the X chromosome we characterized. To my knowledge, this is the first case in which a Hox cluster is located on a sex chromosome, although this PAR region potentially has equal dosage between male and female because of active recombination. Please refer to the individual publications for the rest of the details of our recent achievements. Now I guide you about our ongoing work. Here I list only a subset of species with the reasons of our choice, divided roughly into 2. Sustainable sampling is possible for these species, and these species are accessible in the wild thanks to assistance by aquariums and academic marine research stations. Quite often, we struggle with the frozen samples and are equipped with the tools for cutting off tissue pieces from large frozen bodies uh, for our genomics work. I emphasize that we always try to avoid overlapping effort, and we decided not to proceed with these species because other parties like VGP works on them, if I understand correctly. Including these species, we are working on improved assemblies for or brand new assemblies of various species. The full list is available in our gateway page at GitHub. This is our typical workflow. Now we basically combine PacBio hi-fi read assembly and Hi-C scaffolding. Sometimes we also employ optical mapping with BioNanoSapphire and Oxford Nanopore sequencing. We are now testing ultra-long reads and adaptive sequencing targeting sex chromosomes. Here is a length comparison of vertebrate chromosomes. Sharks and rays often show a gradualism of chromosome length, and they also have quite small chromosomes. But in reality, it is very hard to judge if small scaffold sequences are really chromosomes or they are just fragments of large chromosomes. Even with high-fidelity long reads, some regions are sometimes not sequenced or assembled as expected and remain separate. To mitigate this uncertainty, we are always recommended to obtain independent supports with karyotyping and genome size estimation. Especially, we released a streamlined step-by-step protocol to measure genome size without live cells. We actually lack more hands to complete these experiments for all species we are working on, but we cannot emphasize the importance of these experiments too much. Last but not least, you can access to these small pieces of our experience and products. We are not super proud of these, but some of them are reported in publications, and I hope they enhance the productivity at other parties working on sharks and rays, as well as other groups of species. There are many challenges, but if I focus on common topics to be shared by shark and ray researchers, getting truly reliable sequences is still challenging, and it is important to have genome sequences validated with independent information, namely genome size and karyotype. We need more coordination to efficiently collect data while advancing studies on sharks and rays with minimal sacrifices of wildlife and tackle the whole diversity of chondrixians, and we are keen to contribute to that. Genome-wide sequence data will also be used for various life science studies focusing on individual genes. From this angle, let me also point out here frequent confusions. For example, there are 3 shark homologs of phytologinin genes, but there is no distinction among them in their naming at NCBI protein entries. I introduced this group of genes, phytologinin, earlier in this talk. The hemicerium genome, the epaulet shark genome, was sequenced by the group, including Dr. Manfred Schauto, Dr. Peter Curie, and VGP, but it has remained unannotated at NCBI. Only recently, I communicated with NCBI staff, and they annotated this genome assembly. Probably because of the very high quality, the gene model itself is the best among cartilaginous fishes. However, the labels given to them are sometimes not informative enough. This confusion often occurs when there is no mammalian ortholog. based on our experience. As I said, phylogenes were lost from Eutherian genomes, and this seems to have confused the gene naming logic at NCBI. We feel responsible for this kind of issues as a consortium dedicated to cartilaginous species and are seeking ways to resolve this. At last, let me add these words to welcome your ideas for collaborations and participation in our team. Please contact me if you are interested. Thank you very much for your attention.